introduction. My name is Rubin. So today my topic is about IND enabling CMC strategies for mAb and bispecific mAb. And overall, we want this process as a CMC people. We want to make the CMC process development and manufacturing faster and better. First is about a quick introduction about GenScript ProBio. So GenScript ProBio is a global company with customers from all around the world. In New Jersey, we have a manufacturing facility for plasmid and clinical manufacturing GMP facility. And also in New Jersey, we have a GenScript ProBio headquarters for North America. And for Europe, we have a EU headquarters in Amsterdam. And in China, Nanjing, which is where the GenScript ProBio's headquarters is located. And we have our biologics development, process development, and manufacturing facility in Nanjing. And in Zhenjiang, we're building our phase 3 clinical and commercial manufacturing facility for antibody production. And in Tokyo, we have a ProBio cell office, and in Singapore, we have a GenScript Asia Pacific headquarters. Overall, we are very fast-growing CDMO company. Today, I'm going to focus on the right side of the slides. You can see so far we delivered more than 110 cell line projects for IND filing, and we finished the 71 CMC and CMO projects. And all together, up to now, we got 19 global IND approvals, including the ones from China, FDA, and EMA. We altogether delivered 77 GMP batches and went through 43 customer audits. And the manufacturing we're conducting applies for FDA, EMA, and MDA GMP standard. Most of our project is antibodies and proteins. 46% of all CMC projects were non-mAb proteins, and they're more difficult to develop and more difficult to manufacture. So, for example, 19% of the projects are bispecific and trispecific projects, and 22% of all CMC projects are recombinant proteins. On the right side, I'm showing some of the customers that got IND approval and some of the projects that are ongoing. And on the right bottom, you can see the 5 formats we have been worked before for now. We have SCFE fused with antibody, and we did in vitro assembly for customers, and we worked with knopped in whole formats, and the mAb in cyclotin connection, and also SCFE connected with SCFE formats. For now, we have a 2,600 liter GMP manufacturing capacity that is in Nanjing site. And by the end of this year, actually, we are going to launch our fill and finish line that makes all the drug product and drug substance production in the same site. And by the end of next year, we will have another 16,000 liter manufacturing capability ready. And it will have 8 2,000 liter bioreactors and with fill finish on site. Since host cell line is the start of the CMC projects, I want to talk more about the host cell line, because if you have a right clone or a perfect clone with high productivity, and will help the CMC projects a lot. In GenScript ProBio, we have our CHOK1 genes, wild type cell lines that's licensed from ADCC. And also we developed ADCC plus cell line, which can produce the afucosylated antibody. And both cell lines are adapted to suspension, and they can grow in animal components-free culturing, and they can compatible with the chemical-defined commercial media. In terms of the vector, we also have our in-house vector system, one called DGV system and another one called 1.0 up. Most of the time in our process development, we will do this two vectors in parallel and find the best vector and the host cell line for the client. In normal circumstances, the 1.0 up will have a higher titer for mAb and bispecific antibodies compared with DGV. And for protein projects, we have the 2G unique vector, which we license from Proteonic, which is more specifically for protein production use. In the same time, during the process development, we have our clone ranking and screening platform, which is fully in compliance with FDA, EMA, and NMPA's requirement, and they are high throughput. So in short time, we can screen more clones, which means we will have a higher titer clones. This is a quick summary about how is the productivity of GenScript CHOK1 GS cell line. For mAb projects, on average, we have 5.48 gram per liter. And the highest one so far we got is 9.86 gram per liter in fed batch process. Population cell density. And if you look at the picture on the left bottom, more than 50% of our project final titer was more than 5 gram per liter. And more than even 10% of our project got the final titer more than 7 gram per liter. And there was no any projects from this cell line that has a titer less than 3 gram per liter for monoclonal antibodies. If you look at the right side of the slides, the CHOK1 genes system was also used to express different types of molecules for monoclonal antibody, bispecific antibodies, and recombinant proteins. As I said, for mAb is 4.48 gram per liter on average. And for bispecific antibody, we have on average 3.47 gram per liter. You can see there's a larger variation for the bispecific antibody. It's more because bispecific antibody or trispecific antibody, they have many different formats. So because of the variation of the formats, their titer was also varies a lot. However, if the clients need high titer to reduce the production cost, we can always use the intensified fed batch. Using that intensified fed batch mode, we can double the titer without impacting the product quality attributes and without impacting the CMC progress. And for protein projects, we have on average 2.16 gram per liter. A little bit about ADCC cell line. The reason we develop ADCC enhanced cell line is because it's easiest way and is a way that you don't have to file patents or fight patents with other companies. By using this cell line, you can just having your product ADCC enhanced without worrying any lawsuit or any patent issue, because most of the additives that can increase your ADCC is already filed by some of the competitors in the world. Our ADCC enhanced cell line has a similar growth profile and productivity as the CHOK1 genes cell line. And the product from the ADCC enhanced cell line, you can see on the top of the graph, there's completely no G0F at all because the fucosylation was greatly reduced by using this cell line. And for a case study of the ADCC activity on the left, you can see the affinity to the specific target was increased by 3 to 4 times. And on the middle of the graph, you can see the ADCC activity was increased by 10-fold by afucosylate, the N-glycan modification. And on the right, you can see the EC50 was reduced by 6 times. In conclusion, using this cell line, the ADCC effect was significantly increased. Another common question asked by our clients was, I have 3 or 4 candidates, which one I should choose? And for those needs, we also have our developability assessment. We recommend to do the host cell and the vector system cell pool to produce these target candidates. By doing this, we can keep the product quality consistent between animal studies, developability studies, and CMC studies. Usually, we would pick 3 extreme conditions. For example, high temperature to assess the room storage stability, and we look at the low pH to look at if during the purification if we have any issue, and also we will look at the freeze and thaw to support the shipping and the storage. And by testing the quality profile, we can select the best candidate for following CMC development. In the table on the right, you can see we have a candidate A and B and C. And after all those tests, and after we look at all this purity profile, quality profile, you can see the candidate C would be the best one in practical for CMC development and manufacturing. Last but not least, I want to talk more about the CMC strategy for mAb and bispecific mAb, because we have been doing this for a while, and I want to share some of our experience with other people in this industry. Overall, we have mAb and bispecific mAb platform in ProBio, and for mAb, the key consideration a lot of times are faster timeline, and the clients want to be cost efficiency, and so we would recommend to do the fast CMC trial that's using the fed batch process, and the timeline it takes from DNA to tox only takes 6 months, because once you have the tox batch delivered, the clients can use this product, the produced drug substance for tox study for animal studies. So the CMC process will no longer on the critical timeline after the tox batch is delivered. That's why I specifically list the time from DNA to tox. And for bispecific product, a lot of times the key consideration is whether we can remove the byproducts, and in the same time we want to fully optimize the process, because sometimes bispecific antibody is more fragile depending on the specific formats. And we would recommend either fed batch process or intensified fed batch. It's really depending on the titer needs of the final product. And from the DNA to tox, it takes one more month compared with the mAb, because bispecific antibody needs more downstream tweak compared with the mAb. So from DNA to tox, it takes 7 months. I'm showing a quick timeline for the mAb development. If we go with a fast CMC project, it will take 6 months from the DNA to tox. And as you can see, at the end of month 6, the tox batch will be delivered to the clients for the tox study and in the animals. And it is guaranteed that in the 200-liter batch, we will deliver more than 0.5 gram kilogram product. And in a 500-liter, we will deliver more than 1.2 kilogram product. For bispecific antibody CMC, there are several challenges. For example, low titer and low product stability, and challenges in the purified byproducts. And last but not least, there are also a lot of analytical methods development needs, because sometimes the product and the byproduct are so similar to each other, it's hard to distinguish. So analytical department always have to work harder in order to prepare the specific bioassays to distinguish the byproduct and the target product. For low titer, we always modulate relatively expression of the polypeptides by optimizing the plasmid ratio to reduce mismatch and in the same time improve the titer. And also in upstream and CLD, we have a high throughput screening that can screen the best clone and also screen different media combination. In upstream, we do the process parameter optimization, and we can do intensified fed batch without impacting the product quality attributes. For purification, a lot of times we have to remove the aggregation and mismatch, so we utilize the multi-model resins based on the different types of the impurities, and also you have to tweak different process parameter in order to optimize the purification process. For formulation, is similar to the mAb. You have to evaluate the effects of pH and the buffer system and concentration to the formulation. And we have a lyophilization development capability. So if this product is truly not stable, we can always use a lyophilization process to dry freeze the product, which will have longer shelf life in the future. For analytical development, we would focus on the detection of the homodimers because those are the byproduct that's sometimes hard to remove by downstream. In the same time, we are capable of designing the binding or functional bioassay based on the mechanism of the action. Cell line development, like I just introduced, we would copy the line in different vectors. So by adjusting the vector ratio, we can have a higher titer in the same time, have less aggregation from the clone. And on the right, I'm showing that with Fc region containing bispecific antibody, our titer is on average 3.45 gram per liter. And some of the product is lower than that, and sometimes the product is higher than that. It's more product specific, but on average, the titer is 3.45. And for cell culture process development, it is similar to mAb. There's not too much difference. We have to optimize all the parameters, including temperature, pH, agitation, PCO2, osmolarity, and culture duration. For the culture mode, we have a fed batch process, intensified fed batch process, and perfusion. Most of the time for mAb, fed batch process is good enough to reach a 5 gram per liter titer. And for intensified fed batch, we can use for bispecific antibodies because the titer, if the clients have a high requirement for the titer, we can use intensified fed batch. And if this product, some of the product are not stable in cell culture environment for a long time, so perfusion, we would use perfusion process to address those products. These are some of the cases that we have titer improved in different ways. For example, amino acid supplementation, and on the right you can see pH adjustment also have a quite impact to the productivity. And on the left, on the bottom left, you see that high density inoculation, which is a fed batch process, can really improve the titer by sometimes more than 100%. And also temperature shift is also something that we will test during our process development for upstream. For product quality consideration on the left, you can see some of the product, they're not stable in fed batch cell culture process. So we use perfusion process to exchange those products out after they expressed out of the cell. They are exchanged out of the bioreactor and kept cool. In that case, they can keep stable. And on the right, you can see as the cell culture duration goes longer, we have aggregation come up as the impurities. So in this case, during the process development, we may have to consider the benefit and the concern by prolong the culture duration. And also, we have different ways to improve the glycosylation and cellulation modification. we have to add different supplements, different additives, small molecule additives most of the time. And on the right, if we want to do biosimilar, a lot of times the culture media screening is involved. We will screen different large amount of media to select the best match for the target product. For purification process development, the focus is always byproduct removal. And for aggregation, we have different columns to address those. In the same time, there are fragment and mismatch. We use affinity column, ion exchange column, and multi-model column to address these issues. In this slide, I'm showing a good example of using the multi-model resins. On the left column, you can see the recovery rate. For the traditional cation exchange, the recovery rate is only 15%. And for using the multi-model resin, the recovery rate increased to 70%. And for C column, using the CHT is also a multi-model column. The recovery rate is increased to 75.9%. And the multi-model column have increased the recovery rate, but in the same time, it also have a higher purity. You can look at the SEC profile that's both the resin B and resin C have a higher SEC level, the mAb pattern. For homodimers removing, still on the right graph, you can see for anion exchange column, the homodimer after purification, it still has 38% of the homodimers. And for cation multi-model column, it only have 0.8%. And the product purity on the right, for the traditional cation exchange column is only 61.4%, and for the multi-model resin is reached 99%. For formulation development is more similar to the monoclonal antibody product because still we went through those accelerated conditions to test the rate of aggregation in liquid formulation. And also we have the accelerated degradation that we can find at low pH. So we can determine the formulation based on the aggregation and degradation data. And for analytical method development is always another big part during the CMC development. And each, depending on the impurities, we have analytical group will have to develop a different assay to detect the specific impurities. For example, if the ICIEF is different between the product and the byproduct, we will use ICIEF to distinguish the difference. And if there's the target product and the byproduct have different molecule weight, we will use SDS page. And we can always use LCMS to monitor the undesired pairs, and also the monitoring the process development, also monitoring the manufacture of the final product. For bispecific antibodies, because it has two targets compared with monoclonal antibodies only have one, so it is more tricky to develop the bioassays for two targets. For this one, our bioassay group developed different strategies to verify the mechanism of the action for different target product. If we have two products that is dual immunomodulars, then we will develop two different cell-based assay to separately assess the potency of each antigen target arm of the bispecific antibody. However, if we have the T-cell engager, then we would do a binding ELISA to assess the binding activity of the both antigen, and we will develop one cell-based assay to assess the T-cell activation, as you can see on the the following graph is a good example of this strategy. And ProBio has a well-established bioassay platform that can accurately measure the target MOA for various bispecific antibodies. We have cell-bridging bispecific antibodies, and for dual target blockade, we also have a corresponding cell lines. And for dual immodulars, we have a corresponding cell lines and a target. Because we have our own cell engineering group, this work can be done very quickly. I'm going to provide a quick case for a product. This product is already filed in IND for FDA and NMPA. During the cell line and the upstream process development, we did media screening and amino acid supplement to improve the titer by 62%. And again, we add manganese to lower the MEN5. For purity improve, we tweaked the different process parameters to increase the recovery rate and purity from 84% to 98% for the purity profile. And for bioassay, we selected a suitable bioassay to develop the assay to detect the potency of the multiple targets. This is a case one. So by media screening, we improved the titer by around 40%. And after optimize all the parameters, the final titer was 1.6 times higher compared with the original titer. And because we also, our clients may have MEN5 problem because MEN5 is more than 10%, so we add manganese to reduce the MEN5 to under 10 level. And so for purification, we tweaked, we used different resin and tweaked the different specific parameters. So at the end, both the affinity column and the CX column have improved greatly. For bioassay development, because this is a trispecific antibody, it is harder. What's the final strategy was we have two bioassay. The first cell assay is targeting the target 1 and target 3, and the second bioassay was targeting target 2 and target 3. By doing this, we can verify both the mechanism of this trispecific antibody. The case study 2 is an in vitro sample of the bispecific antibody. For this one, we have the target 1 connected with the linker first, and the second part of the SCFV was connected to the linker. And in the third step, both SCFV part was connected together and the linker was removed. In this step, again, we optimize the linker conjugation for SCFV1 and 2 in the first place, and after all, we optimize the final conjugation step. So the ratio of the recovery was improved from 29% to 64%. That's probably near the end of my presentation. So in conclusion, ProBio has a leading bispecific antibody CMC platform, which can break through many CMC barriers. We have a high expression system. We have a good yield. In the same time, we have a strong analytical assay platform. And our CHOKI-1 genes and the CHOKI-1 ADCC system can provide the clients world-class productivity and also ADCC enhancement. And ProBio has a well-established bioassay platform that can accurately measure the target of MOA for various bispecific antibodies. For a lot of companies, they may have here because bioassay platform development is always time-consuming, and there are uncertainties in the assay development. However, in ProBio, because we have our own cell engineering group, it's much easier and for us to produce a robust bioassay platform. With this, I'm ending today's presentation, and I'm looking forward to the questions. Thank you a lot.
Thank you, Mr. Jiang, for the great presentation. So now let's